Hello, good evening, madam.
Hello, sir. Good evening.
Uh, yeah, madam.
Uh, can we continue, sir?
Yeah, please continue, madam.
Okay, sir. So, hello everyone. Welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class is about systemic bacteriology, part 1 for BSc MLT second year students. So, introduction to systemic bacteriology. Systemic bacteriology deals with bacteria causing infections that affect multiple systems or organs through systemic spread via bloodstream or lymphatics. The subject aims to study bacteria's morphology, physiology, biochemical characteristics, and pathogenic mechanism to aid in disease diagnosis and treatment. The understanding of systemic infections helps clinicians manage severe infections like septicemia and meningitis effectively. So, why we study systemic bacteriology? It is important because it allows identification of bacterial pathogens that cause infections affecting entire body systems rather than localized sites. So, this knowledge helps clinicians diagnose serious infections such as septicemia, pneumonia, meningitis, and endocarditis. So, accurate identification supports targeted treatments with appropriate antibiotics, reducing resistance development. It also contributes to epidemiological surveillance by tracking outbreaks and infection control. For laboratory workers, it builds foundational skills in culture techniques, stainings, and biochemical testings. So, theory with practical diagnostic and research. So, biochemical tests are used to identify and classify bacteria based on their metabolic pathways and enzyme presence. Key tests include catalase, coagulase, IMVIC, urease, oxidase, gelatin, liquefaction, nitrate reduction, and sugar fermentation. These tests examine bacteria's ability to break down substrates like hydrogen peroxide, plasma, sugars, urea, and nitrates, which can differentiate between bacterial species and identify pathogenic from non pathogenic strains. Motility test also helps identify if bacteria possesses flagella and are mobile. So, what is the scope and focus of the study? It includes a detailed bacterial classification based on morphology, that is, shape, size, and arrangement, physiology, growth requirement, oxygen tolerance, and metabolism, which is enzyme production, fermentation capabilities. Key focus areas are recognizing pathogenic species that cause systemic illness, understanding their virulence factors, which is toxins, adhesions, immune invasions, and mastering laboratory methods such as culture, microscopy, staining, and biochemical tests. So, the study links clinical symptoms with laboratory results to provide comprehensive bacterial diagnosis. So, these enzymatic and metabolic pathways differences among bacteria. So, different bacteria produce unique sets of enzymes and have specific metabolic pathways. Some produce catalase, which decomposes hydrogen peroxide, while others produce urease, breaking down urea to ammonia and carbon dioxide. Sugar fermentation patterns vary across species, as do abilities to reduce nitrate and liquefy gelatin. These biochemical differences form a fingerprint utilized to differentiate bacterial species in the lab underlying biochemical test selection. So, uh, we'll go one by one. These are the list of tests covered, which is the major biochemical test: catalase test, coagulase test, IMVIC test, which is indole, methyl red, Vox, proscure citrate, urease test, oxidase test, gelatin liquefaction test, nitrate reduction test, sugar fermentation test, motility test, and last is oxidase fermentation test. Catalase test, why we do catalase test? To detect catalase enzyme. Coagulase test detects coagulase enzyme, and these will detect indole, methyl red, Vox, and citrate test. Urease will detect urea enzyme activity. Oxidase will detect cytochrome oxidase enzyme. Gelatin liquefaction test will detect gelatinase enzyme. Nitrate reduction test will detect nitrate to nitrite reduction. Sugar fermentation test, it will detect acid gas from carbohydrate metabolism. Motility test detects bacterial motility. And last, oxidase fermentation, it will differentiate aerobic oxidation and anaerobic fermentation. We'll go one by one. Catalyst test. So the catalyst test detects the presence of the enzyme catalase, which is produced by aerobic and facultative anaerobic bacteria to neutralize hydrogen peroxide, a toxic reactive oxygen species. Catalase catalyzes the decomposition of hydrogen peroxide into water and oxygen, which is H2O and O2. So importance is differentiation. These tests distinguishes catalase positive bacteria such as Staphylococcus, Staphylococcus bacteria, Micrococcus, and Bacillus species from catalase negative Streptococcus and Enterococcus species. Aerobic versus anaerobic, it helps differentiate aerobic and facultative anaerobic bacteria from obligate anaerobes. Identification used in clinical laboratory as an early step for bacterial identification, and special uses helpful in identifying organisms in the genus Mycobacterium and differentiating Bacillus, which is catalase positive, from Clostridium, which is catalase negative. Materials required will be fresh bacterial culture, preferably 18 to hours old to avoid false result. 3% hydrogen peroxide solution, sterile glass slide, sterile inoculating loop or wooden stick, protective equipment which is gloves, eye and protection. Method, there is two method. First is slide method and the another one is tube method. So slide method will place a small drop of 3% hydrogen peroxide on a clean glass slide using a sterile loop or wooden stick. Transfer a small amount or portion of bacterial colony, not including agar, and mix gently in the hydrogen peroxide drop. After that, observe immediately for bubble formation. For tube method, place 1 or 2 ml of 3% hydrogen peroxide in a test tube using a sterile loop. Pick bacteria from culture and immerse in the hydrogen peroxide. Observe for effervescence of oxygen bubbles. Here precaution is avoid contamination with bubble agar when picking colonies, as red blood cell will contain catalase which may cause false positive result. Use fresh cultures because older cultures may lose enzyme activity causing false negativities. Platinum loops can catalyze hydrogen peroxide decomposition independently, so wooden or nichrome loops are preferred. And perform the test in a biosafety cabinet or with PPE to limit aerosol inhalation of catalase positive organism. Observation and result interpretation. Positive reaction, rapid and vigorous bubbling indicating catalase enzymes presence, which is Staphylococcus. Negative reaction, no or minimal bubbling indicating absence of catalase, which is example is Streptococcus. Weak positive, few bubbles or delayed bubbling considered weak positive, interpreted cautiously. So quality control. Here, positive control, Asorius culture should show bubbling. Negative controls, Streptococcus pyogenes culture should show no bubbling. Next is coagulase test. The coagulase test identifies the ability of certain Staphylococcus species, notably Staphylococcus aureus, to produce the enzyme coagulase. Coagulase reacts with plasma fibrinogen to convert it into fibrin, causing clotting of the plasma. This clot formation is used to differentiate pathogenic S aureus, which is coagulase positive, from other non-pathogenic staphylococci, which is coagulase negative. There is two forms of coagulase which is recognized. bound coagulase, which is clumping factor. It is attached to the bacterial cell wall. It reacts with fibrinogen, causing bacterial cells to clump, detected by the slide coagulase test. And second is free coagulase, which is secreted enzyme that reacts with a coagulase reacting factor, that is CRF, in plasma to produce clotting, detected by the tube coagulase test. Procedure: slide coagulase test. Place 2 drops of saline on a clean slide. Emulsify a colony of the bacterium in the test saline. Drop to form a smooth suspension. Add drop of citrated rabbit or human plasma to the suspension and mix gently. Observe for visible clumping or agglutination within 5 to 10 seconds. Clumping indicate positive bound coagulase. The control saline drop should show no clumping to rule out auto-agglutination. Tube coagulase test. Here you dilute plasma with saline, usually 1:10 ratio. Add 0.5 ml of diluted plasma to test tubes labeled test positive control and negative control, and then add 0.1 ml of an overnight broth culture of test bacterium to the test tube. To the positive control tube, add a known coagulase positive Staphylococcus aureus culture. To the negative control, add sterile broth. Incubate tubes at 37 degrees Celsius and observe for clot formation at intervals up to 4 hours. If no clot forms after 4 hours, incubate overnight at room temperature. Clotting indicates free coagulase positivity. Observation and timing interpretation. Clumping on slide, positive bound coagulase indicate SRS. Clot formation in tube, positive free coagulase confirming pathogenic SRS. No clumping or clotting, coagulase negative staphylococci. Importance: clinical for diagnosing infections caused by SRS. Differentiate pathogenic staphylococci from harmless species, and it will help predict virulence since coagulase enhances bacterial defense against host immunity. So, precaution slide test is rapid but less sensitive. Negative slide should Negative slide test should be confirmed by tube test. Handle plasma and bacterial cultures carefully to avoid contamination. So plasma clotted, it will indicate coagulase positive, pathogenic staphylococcus. Plasma not clotted, it will indicate coagulase negative, which is non-pathogenic staphylococcus. Next is IMVIC test, which is indole, methyl red, Vox proscure, and lactis citrate. So we'll start with indole. The indole test detects the ability of certain bacteria to enzymatically degrade the amino acid tryptophan into indole, pyruvic acid, and ammonia. This breakdown is catalyzed by the enzyme tryptophanase. This tryptophan. So, indole production is a key biochemical feature used to differentiate members of the Enterobacteria family. Bacteria such as E. coli, Prox vulgaris, and Morganella morgani typically test positive for indole production. Indole combines with reagents, which is coax, to produce a color change visible in the test medium. The test is important for rapid presumptive identification of clinically significant pathogens. Here, method use is 48 hours old culture in peptone or tryptophan water. Add indole reagent to see the result. So, procedure: bacteria inoculated into tryptophan broth, incubated for 18 to 24 hours or 48 hours at 37 degrees Celsius to allow tryptophan degradation. After incubation, coax reagent is added, forming a red or pink color layer on the broth surface. Within 1 minute, if indole is present, you'll get red or pink ring. If indole is present, alternatively, a spot indole test can be done by applying bacterial colonies onto filter paper saturated with indole reagent. Positive results show blue to green coloration. Use of enriched reagent is preferred for non-fermenters or anaerobes. So, result and interpretation. Positive, formation of red or pink ring on the broth surface after adding coax reagent or blue-green color on filter paper if spot indicate the presence of indole. Indole positive bacteria include E. coli, Proteus, and Morganella. Negative, no color change or yellow or oily layer without pink ring indicate absence of indole production. Example is Klebsiella pneumonia or Enterobacter species. Principle behind this indole test is methyl red. Next is methyl red. Principle behind methyl red. The MR test differentiate bacteria based on their ability to produce stable acid end products from glucose fermentation, lowering the pH of the medium. Bacteria performing mixed acid fermentation produce a significant amount of acid such as lactic acid, acetic acid, and formic acid that stabilize the low pH. The test uses methyl red indicator, a pH indicator that turns red at pH less than 4.4, which is acidic, and yellow at pH larger than 6.0, which is neutral or basic, neutral to basic. Procedure for MR test. Here you inoculate glucose phosphate peptone broth with bacterial culture and incubate for 48 hours at 37 degrees Celsius. Add methyl red indicator to the culture broth. Positive test, red color indicates stable acid production and positive mixed acid fermentation. Example is E. coli and Proteus species. Negative test, yellow color indicate less acid production and negative mixed acid fermentation. Example is Enterobacter and Klebsiella. Next is Vaux-Proscore VP test. The VP test detects bacteria producing acetyl methyl carbinol, a neutral end product from glucose fermentation. Acetone reacts with reagents alpha naphthol and potassium hydroxide KOH under aerobic conditions to produce a red or pink color. Procedure involves adding this reagent to a 2 to 5 day old glucose phosphate peptone broth culture and observing the color change within 30 minutes. So principle behind this test, the next, which is a citrate test. Citrate test determines the ability of bacteria to utilize sodium citrate as the sole carbon source and ammonium salt as the sole nitrogen source. Bacteria possessing the enzyme citrate permease can transport citrate across their cell membrane. Citrate is metabolized to oxaloacetate and then further broken down into pyruvate and carbon dioxide, which is CO2. The metabolism of ammonium salts produces ammonia, raising the pH of the medium. The resulting alkaline pH changes the color of the pH indicator bromothymol blue in the Simmons citrate agar from green to blue. Green is neutral, blue is alkaline. Materials and medium. Simmons citrate agar contains sodium citrate as the sole carbon source, ammonium dihydrogen phosphate as the nitrogen source, and bromothymol blue as the pH indicator. So method procedure using a straight wire, lightly streak a well-isolated bacterial colony on the surface of the Simmons citrate agar slant. Incubate aerobically at 35 to 37 degrees Celsius for about 24 to 48 hours. Some bacteria may require up to 7 days. Observe for bacterial growth on the slant and monitor the color change of the medium. Interpretation. Positive test growth on the citrate agar with a color change to blue indicates utilization of citrate and production of alkaline products. Example is Klebsiella species, Enterobacter species, and Salmonella species. Negative test, no growth or color remains green, indicating inability to utilize citrate. So no color change will happen. Example, E. coli, Shigella, and Salmonella. This test is critical component of the MIVIC series and aids in the identification of Enterobacteriaceae family members in clinical microbiology. So here is the whole summary table for this test. Genus is E. coli, Enterobacter, Klebsiella, Salmonella, and Proteus. For indole, E. coli is positive, MR methyl red is also positive, VP is negative, and citrate is negative. For Enterobacter, indole is negative, MR methyl red is negative, VP is positive, and citrate is positive as well. For Klebsiella species, indole is negative, MR is negative, VP is positive, citrate is also positive. Salmonella, indole shows negative result, MR shows positive result, VP is negative, and citrate is positive. For Proteus, indole is positive, MR is positive, VP is negative, and citrate is positive. Next is a urease test. The urease test detects urea hydrolysis into ammonia and carbon dioxide by urease enzyme, raising pH and changing medium color, often pink in urea agar. Proteus species are classic urease positive bacteria. These tests aids in diagnosing urinary tract infections and differentiating bacterial species. Methods and results. It will inoculate heavily onto Christensen's urea agar slant and then incubate at 37 degrees Celsius. After incubation, examine 4 hours later and then daily for up to 5 days. Pink or reddish pink will indicate urease positive. Next is oxidase test. Oxidase test identifies presence of cytochrome oxidase enzyme crucial in electron transport of aerobic respiration. Bacteria treated with oxidase reagent turn dark purple to black if positive. Example is Pseudomonas and Neisseria. Negative organism example is Enterobacteria ci. Do not change color, helps differentiate pathogens efficiently. Method: use colonies from agar plate, apply oxidase reagent example para amino dimethyl aniline monochloride. Within few minutes, positive colonies will change color. It will show pink or red to black. Enterobacteriae generally oxidase negative. Pseudomonas neisseria are often positive, oxidase positive. Next is gelatin. It will detect gelatinase enzyme that liquefies gelatin. Bacteria inoculated in gelatin medium, incubated and afterward refrigerated. Liquefaction indicate positive test. Example Serratia, useful and in distinguishing bacterial species with proteolytic activity. Methods and result: inoculate gelatin swab medium and then incubate it for almost up to 48 hours with uninoculated control and then refrigerate until control solidifies. If test sample does not solidify, it will indicate gelatin liquefaction is positive. Next is nitrate. Nitrate tests ability to reduce nitrate, that is NO3 to nitrite, which is NO2, or other nitrogenous compounds anaerobically. After incubation, reagents sulfonalic acid and alpha naphthalamine added cause red color if nitrite present, which is positive. Zinc added if no red color. If red, then negative. Helps in identifying facultative anaerobes and differentiation. So, grow in nitrate broth, add reagent A, which is sulfonalic acid, plus reagent B, which is alpha naphthalamine. If red color will appears, it means nitrate is present and nitrite is positive. If no red color indicate, add zinc. Red after zinc, it will indicate negative, which is nitrate still present but no red color at all. Reduction beyond nitrite. Next is sugar fermentation. Sugar fermentation test assess the ability of bacteria to ferment specific carbohydrates, which is glucose, lactose, sucrose, and maltose, to produce acidic or gaseous end products. Products of fermentation, which is acid or gas, change the pH of the medium, typically indicated by color changes in pH indicator like phenol red. These tests helps differentiate bacteria based on their characteristic sugar fermentation profiles. Motility test hanging drop, which is microscope or semi-solid media with TTC to detect movement, oxidation, fermentation, semi-solid medium with glucose and bromothymol blue, one tube aerobic and another is sealed, which is anaerobic. So here, interpretation: positive acid production, the broth color change from red to yellow, indicating acid production, lowering pH. Gas production, presence of gas bubbles or displacement of medium in Durham tube, indicating gas production. Negative result, no color change. So and gas indicate inability to ferment the sugar. Next topic is antimicrobial susceptibility, which is AST. Antimicrobial susceptibility testing is one of the most critical aspects of clinical microbiology diagnostics. AST helps determine which antibiotics a bacterial pathogen is sensitive or resistant to, directly influencing patients' treatment and outcomes. So, introduction to antimicrobial drugs. Antimicrobial drugs are substances that kill or inhibit the growth of microorganisms. These agents are classified based on their target organisms. Antibacterials target bacteria, antifungals target fungi, antivirals target viruses, and antiparasitics target parasitic infections. So these antibacterial, antifungal, antiviral, and antiparasitic infections, this classification helps guide therapeutic use according to the type of infections. Origins and classification, historical background. So here the slide outlines the history of antimicrobial drug discovery, highlighting Paul Ehrlich, his early work with arsenic-based compounds, which is a German scientist. Despite toxicity, marked the beginning of antimicrobial research. The revolutionary discovery of penicillin by Alexander Fleming in early 1928 ushered in the antibiotic era, dramatically changing infectious diseases management. So these two scientists, first, the development of antimicrobial drugs began in the late 1800s with Paul Ehrlich, a German scientist who discovered that arsenic compounds were an effective treatment for syphilis. Unfortunately, these compounds, arsenic, were also highly toxic to the patients they were being used to treat the infection. In early 1928, Sir Alexander Fleming accidentally stumbled upon the discovery of the wonder drug penicillin, which is the most commonest drug nowadays. As he was inspecting a plate of Staphylococcus aureus contaminated with the mold penicillium, Fleming noticed that the mold had inhibited the growth of the bacterial colonies. He later isolated the compound responsible for this inhibition and named it the drug penicillin. Classification of antimicrobial drugs. It will be based upon how they are made and based upon their spectrum or range of effectiveness and their primary site of infection. So antimicrobials are broadly classified based on their source: natural products, products produced by fungi or bacteria; semi-synthetic, semi-synthetic. So semi-synthetic will be chemically modified natural drugs, and fully synthetic, chemically synthesized in the lab. We'll go one by one. We'll start with naturally derived antibiotics. Natural antimicrobials such as penicillin from penicillium have long been used. Here, cephalosporins comes from the fungus Cephalosporium, penicillin derived from the fungus Penicillium, bacteriocin and polymyxin, which are produced by the soil bacteria, Bacillus licheniformis and Bacillus polymyxa. Next is semi-synthetic drug. Semi-synthetic drugs like ampicillin improve stability and spectrum of natural drugs. It will isolate from natural sources but then chemically modified in a laboratory to make the drugs more effective, long-lasting, easier to administer, and which is less toxic to the patient's health. Example is again ampicillin and methicillin are semi-synthetic drugs derived from penicillin. Next is synthetic drug. Synthetic antimicrobial design entirely in the lab offer the advantage of tailoring properties but may carry increased risk of toxicity or release resistance and development. Classification based upon their spectrum. So spectrum of activity. Broad spectrum antibiotics are effective against many bacterial species, including gram-positive and gram-negative organisms, but may disrupt normal host flora and lead to selection of resistant organisms. Narrow spectrum antibiotics are targeted against specific pathogens, minimizing collateral damage and resistance pressures. Next is, so here is the spectrum of antibiotics and other antimicrobial drugs for prokaryotes and eukaryotes and viruses. For prokaryotes, mycobacteria, gram-negative bacteria, gram-positive bacteria, and chlamydiasis, rickettsiasis. These image you can refer for the spectrum of antibiotics and other antimicrobial drugs. For eukaryote, protozoa, fungi, and helminthes, and for viruses, eridone, ribavirin, and acyclovir. Next is classification. So this slide likely uses charts or diagrams to visually represent antibiotic effectiveness across various bacterial groups, highlighting clinical relevance in drug selection. Next is classification. Primary mode of action. Antimicrobial drugs, antibacterial drugs regulate microbial growth and division by inhibiting cell wall synthesis, disrupting the plasma membrane, blocking protein synthesis, disrupting metabolic pathways, and inhibiting nucleic acid, which is either DNA or RNA synthesis. So, cell wall inhibitors, penicillin, cephalosporin, bacitracin, and vancomycin interfere with peptidoglycan synthesis essential for bacterial cell wall strength. These drugs are typically bactericidal and effective mainly against actively dividing bacteria. Next is membrane disruptors. So here polymyxins interact with polysaccharides and phospholipids in gram-negative bacterial membranes, altering permeability and causing cell death. Due to toxicity, polymyxin are reserved for multi-drug resistant infections. Next is blocking protein synthesis. Drugs targeting the ribosomal subunits impair bacterial translation mechanism. Aminoglycosides and tetracyclines bind the 30S subunit, causing misreading or blocking tRNA attachment. Macrolides and chloramphenicol binds the 50S subunit, preventing peptide bond formation. Next is disrupting metabolic pathways. So, metabolic pathways, sulfonamides and trimethoprim inhibit enzymes in the folic acid pathway, which is necessary for nucleotide synthesis, thereby arresting bacterial growth. Inhibiting nucleic acid, DNA or RNA, fluoroquinolones inhibit DNA, gyrase and topoisomerase, essential for DNA replication. Rifampin interrupts bacterial RNA polymerase activity, inhibiting RNA synthesis. So one by one, we'll see all these cell wall inhibitors. It will block the synthesis and repair of the bacterial cell wall and include drugs such as penicillin, cephalosporin, vancomycin, and bacitracin. Plasma membrane inhibitor. It is a drug that disrupts the bacterial plasma such as polymyxin. Protein synthesis inhibitors, it will include those that target the 50S ribosomal subunit, which include chloramphenicol, erythromycin, and clindamycin, and those that target the 30 ribosomal subunit, which is aminoglycoside, tetracyclines, and streptomycin. Nucleic acid inhibitor block replication and transcription and include drugs such as ciprofloxacin, DNA, and rifampin, which is RNA. Metabolic inhibitors, it will prevent folic acid synthesis and include sulfur drugs and trimethoprim. Drug resistance. Drug resistance. Development of drug resistance emerges through spontaneous mutations or horizontal gene transfer mechanisms such as conjugation, transformation, and transduction. Improper use of antibiotics accelerates resistance spread, threatening global health. So here, microbes develop drug resistance due to genetic mutation and by acquiring genes from other microbes via processes like this conjugation, transformation, and transduction, as I mentioned earlier. Humans contribute to the development and spread of drug resistance by these three terms: not using the appropriate drug for a particular type of infection, not finishing medication or not taking it properly, using antimicrobial drugs when they are not needed. So these three things is important. When an antimicrobial drug is present and the majority of drug sensitive cells die, the drug resistant cells left behind will be allowed to grow unchecked until the outer number of the drugs sensitive cells will form. Here is an image diagram of drug resistance. So it is divided into three parts. A population of microbial cells. Here you can see drug sensitive cells and drug resistant mutant. Once when they expose to drugs, here sensitive cells will be inhibited by exposure to drugs, and then later remaining population will overgrow over the period of time. Now here in last stage, more cells are now resistant. So why it is important? Multi-drug resistant, it will fail to respond to the standard treatment, resulting in prolonged illness and greater risk of death. Patients remain infectious for a longer period of time, increasing the risk of spreading resistant microorganisms to others. Drug resistance leads to treatment failures, increased mortality, prolonged hospital stays, and increased healthcare costs. It also facilitates the emergence of untreatable infections. Some types of bacteria have developed multi-drug resistant forms. Example is S. aureus have developed resistance to penicillin, methicillin MRSA, which is methicillin resistant Staphylococcus aureus, and vancomycin, which is vancomycin resistant Staphylococcus aureus. Definitions of MIC, minimum inhibitory concentration, MBC, minimum bactericidal concentration. So these key terms of AST. The minimum inhibitory concentration is the smallest drug concentration preventing visible bacterial growth and is used to define susceptibility. The minimum bactericidal concentration is the smallest drug concentration killing a defined percentage, often 99.9% of bacteria. Susceptible based on determined MIC values. Bacteria are classified as susceptible, which is treatable with standard dosage, intermediate, possible clinical efficacy at higher doses, or resistance, drug likely ineffective. So susceptible interpretive category that indicates an organism is inhibited by the recommended dose at the infection site of an antimicrobial agent. Intermediate interpretive category that represent an organisms that may require a higher dose of antibiotic for a longer period of time to be inhibited. Resistance interpretative category that indicates an organism is not inhibited by the recommended dose at the infection site of an antimicrobial agent. So, purpose of AST facilitates rational antibiotic selection by determining in vitro susceptibility, thereby improving patient outcomes and reducing unnecessary drug use. When to perform AST? AST is indicated when pathogens are isolated from sterile or infected sites. Infection is severe or unusual when treatment failure occurs, especially in immunocompromised patients. Selecting antibiotics for testing. So, here factors: body side of infection, susceptibility not performed on bacteria, isolated from body side where they are normal. Flora, example is susceptibility for E. coli is not performed when isolated from stool, but IS is performed when isolated from blood. Presence of other bacteria and quality of specimen, example is two or more organisms grown in a urine specimen. Host status, it will depend on immunocompromised patients or allergies to usual antimicrobials. Next is selecting antibiotics for testing. Selection prioritizes drugs commonly used clinically, considering spectrum, drug availability, toxicity, and local resistance patterns, ensuring relevant and active notable results. So, Clinical and Laboratory Standard Institute CLSI is must develop standards, methods, QC parameters, and interpretive criteria for sensitivity testing. If necessary, can alter the breakpoints of the SIR, which is susceptible, intermediate, and resistant based on emerging resistance. There are approximately 50 antibacterial agents. Follow CLSI recommendation. Each laboratory should have a battery of antibiotics ordinarily used for testing. Drug formulary decided by medical staff, pharmacists, and medical technologists. So the selection of antimicrobial agent, generally lab will choose around 10 to 15 antibiotics to test susceptibility for organism and another 10 to 15 for another organism. Too many choices will confuse physicians and be too expensive. Primary objective is use the less toxic one, most cost effective and most clinically appropriate agents, which is cheaper in cost and broad spectrum agents. Next is ideal antimicrobial agent. An ideal antimicrobial medication should be highly toxic to the microbe, non-toxic to the host, not interfere with the ability of the host to fight with other diseases, and last but not the least, it should not lead to the development of drug resistance. Methods of performing AST: agar dilution method. Agar dilution method, serial antibiotic dilutions are incorporated into agar plates. Each plate is inoculated with fixed bacterial inoculum spots and inspected after incubation for growth inhibition zones to define MIC. Broth macrodilution or tube dilution. Antibiotics here diluted in broth form a series of concentrations, bacterial inoculum added and incubated. Visible growth or turbidity assessed. Microbroth dilution uses microplate allowing high throughput. This diffusion method. Standardized bacterial suspensions are evenly spread on Mueller-Hinton agar plate. Antibiotic discs impregnated with fixed doses are placed. Zone of inhibition measured and interpreted using CLSI criteria. Gradient method. Here, plastic strips infused with a gradient of antibiotics are placed on inoculated agar plates. The MIC is read where the elliptical inhibition zone intersects the stripes concentration scale, providing a quantitative results. Next is standardization. So inoculum preparation, use of 4 to 5 colonies, not just one colony. Inoculum standardization is must and using McFarland's 0.5 standard for susceptibility testing. These are the methods as I mentioned earlier: agar dilution, broth, macro dilution, tube dilution, diffusion, and gradient diffusion. So for agar dilution, dilutions of antimicrobial agent added to the agar plate and growth on agar indicate the presence of MIC. Broth, macro dilution, and tube dilution, two-fold serial dilution series, each with 1 or 2 ml of antimicrobial. It will be too expensive and time-consuming. Micro dilution test, plastic trays with dilution of antimicrobial will be used. Disk diffusions by Kirby-Beer method. So Kirby-Beer method, standardized bacterial suspensions are evenly spread on Mueller-Hinton agar plate. Antibiotic disc impregnated with fixed dosage are placed here. Zone of inhibitions measured and interpreted using CLSI criteria. So procedure is use a well isolated around 18 to 24-hour old organism, transfer organisms to a broth, either tryptic soy or sterile saline, ensure a turbidity using 0.5 McFarland method, inoculate image agar by swabbing in 3 different directions, lawn of growth, place a filtered paper disc impregnated with antimicrobial agents on the agar plate, invert and incubate for 16 to 18 hours at 35 to 37 degrees Celsius in non-carbon dioxide. Please, during incubation, drug diffuses into agar, and depending on the organism and drug, areas of no growth form a zone of inhibition. As you can see in this picture, zones are measured to determine whether the organism is susceptible, intermediate, or resistant to the drug. Next is E-test, which is by gradient or diffusion method. It will be done on MIC stick. Plastic strips are impregnated with the antimicrobial on one side, MIC scale on the another side, and then read MIC where zone of inhibition intersects E strip scale. As you can see in this picture, this is called as E stripe, and it will be placed on an agar plate. After incubation, zones will be measured and compared to the standard one. Automated EST system. Systems like Vitek-2 and Phoenix automate broth microdilution and use optical or fluorometric detection for rapid standardized susceptibility reading, shortening turnaround times in diagnostics. So it will detect microvolumes of broth with various dilutions of antimicrobials, detection via photometric, turbidimetric, or fluorometric method. Types include BD Phoenix, Microscan Walkaway, Trex and C Titer, Vitek 1 and Vitek 2. Advantages and disadvantages of using automated AST system. Advantages is increased reproducibility, decreased labor costs, a rapid result, software will be included, detects multi-drug resistant organism, ESBLS correlates bacterial ID with sensitivity. Disadvantage is it is cost effective. Quality control in susceptibility testing. So quality control involves testing with references strains ATCC. These reference strain ATCCs to validate procedures. Superbugs such as MRSA, VRE, and CRE show resistance to multiple drugs, necessitating strict infection control, stewardship programs, and ongoing susceptibility surveillance. So here it reflects types of patient isolates and ranges of susceptibility. Frequency of quality control depends on methods according to CLSI guidelines or manufacturer. Reference strains of QC material, American type culture collection, which is ATCC, E. coli ATCC given 25922 and S. aureus ATCC is 25923. The superbugs. Organism resistance to previously effective drugs is known as superbugs. MRSA and methicillin resistant Staphylococcus aureus. MECA genes code for a PBB that does not bind to beta-lactam antibiotics. It is resistant to oxacillin, vancomycin, and VRE enterococcus species and Staphylococcus aureus. The superbugs beta-lactamases. Here, gram-negative rods that have genes on chromosomes will code for enzyme against certain antimicrobials. It will show extended spectrum beta-lactamases and is resistant to extended spectrum cephalosporins, penicillins, and azithromycin. Examples include E. coli, which is Escherichia coli and Klebsiella species, Carbapenemesis, Klebsiella pneumonia, resistant to it. This organisms is resistant to penicillin, cephalosporins, carbapenems, and it will inducible organisms. Controlling the superbugs, lab's role is recognize and report isolates covered from clinical specimens. Methods for identification include automated systems, screening agars, and role of health and co-workers, hand hygiene with use of sterile alcohol-based hand rubs or soap or and water after patient care, performing lab procedure. So sterilization is must. Contact precautions from patient identified as colonized or infected with superbugs. Healthcare personnel education about the method of transmission, how to handle it, contact precautions, and proper use of hand hygiene. Minimization of invasive devices, which is catheters, etc. Proper administration of antimicrobial agents where therapy is selected for susceptible organisms for the proper duration. Next is normal flora. Normal flora, the mixture of organisms regularly found at any anatomical site, is referred to as the normal flora. The normal flora of human is exceedingly complex and consists of more than 200 bacterial species. The makeup of the normal flora depends upon various factors including genetics, age, sex, stress, nutrition, and diet of the individual. The normal flora of human consists of a few eukaryotic fungi and protists and some methanogenic archaea that colonize the lower intestinal tract. But the bacteria are the most numerous and obvious microbial components of the normal flora. Importance of normal flora. Bacteria are the most numerous and obvious microbes. It provides nutrients, protects against pathogens, stimulate immunity, and it will colonize and adapt to host tissues via adhesins and receptors. Next is host microbe relationship. Mutualistic relationship. The normal flora derives from the host a supply of nutrients, a stable environment, and constant temperature, protection, and transport. The host obtains from the normal flora certain nutritional benefits, stimulation of the immune system, and colonization strategies that exclude potential pathogens at the site. So, the normal flora are obviously adapted to their host tissues, most probably by biochemical interactions between bacterial surfaces, components, ligands or adhesions, and host cell molecular receptors. So in general, there are three explanations for why the normal bacterial flora are located at the particular anatomical sites. First is the normal flora exhibit a tissue preference or prediction for colonization. This is referred to as tissue tropism. One's explanation for tissue tropism is the host provides an essential growth factor needed by the bacterium. Of course, to explain why bacteria are not an alternative site, the host internity provides an inhospitable environment for the bacterium by the production of such substances as stomach acids, bile salts, and lysozyme. Second is many of the normal flora are able to specifically colonize a particular tissue or surface using their own surface components like capsules, fimbriae, cell wall components as specific ligands for attachment to specific receptors located at the colonization sites. Third is some of the indigenous bacteria are able to construct bacterial biofilms on a tissue surface, or they are able to colonize a biofilm built by another bacterial species. Many biofilms are a mixture of microbes, although one member is responsible for maintaining the biofilm and may predominate. So the composition of the normal flora, it has been calculated that the normal human is host to about 10 to the power of 12 bacteria on the skin, 10 to the power of 10 in the mouth, and 10 to the power 14 in the GI tract, that is gastrointestinal tract. We'll start with normal flora of skin. An adult is covered with approximately 2 square meters of skin. The density and composition of the normal flora of the skin vary with anatomical locale. The high moisture of content of the axilla, the groin, and the skin between the toes and near orifice supports the activity and growth of relatively high densities of bacterial cells. But at most other sites, the bacterial population is fairly low, generally in hundreds or thousands per square centimeter. The majority of skin microorganisms are found in the most superficial layers of the epidermis and the upper parts of the hair follicles. So they consist largely of Asorius epidermis and Micrococcus species and Corynebacteria. These species of bacteria, these are generally non-pathogenic and considered to be commensal, although mutualistic and parasitic roles have been assigned to them. For example, skin bacteria or their metabolites may protect against colonization by dermatophytic fungi, or bacterial metabolites on the skin may contribute to the development of certain skin cancers. Sometimes potentially pathogenic Asorius is found on the face and hands, particularly in individuals who are nasal carriers. Qualitatively, the bacteria on the skin near any body may be similar to those in the orifice. So this is the normal flora of skin. Next is respiratory tract. The nares, which is nostrils, are always heavily colonized, predominantly with Asorius epidermis and Corynebacteria, and often about 20% of the general population, with Staphylococcus aureus, this being the main carrier site of this important pathogen. The healthy sinuses, in contrast, are sterile. A large number of bacterial species colonize the upper respiratory tract, which is nasopharynx, and includes predominantly non-hemolytic and alpha-hemolytic streptococci and Neisseria species, and sometimes pathogens such as Streptococcus pneumonia, influenza, and Neisseria meningitis. So the lower respiratory tract, which is trachea, bronchi, and pulmonary tissues, is virtually free of microorganisms, mainly because of the efficient cleansing action of the ciliated epithelium, which lines the tract. Any bacteria reaching the lower respiratory tract are swept upward by the action of the mucociliary blanket that lines the bronchi, to be removed by subsequently by coughing, sneezing, swallowing, etc. If the respiratory tract epithelium becomes damaged, as in bronchitis or viral pneumonia, the individual may become susceptible to infection by pathogens descending from the nasopharynx. Example is Haemophilus influenza or pneumonia. Next is oral cavity. The presence of nutrients, epithelial debris, and secretions makes the mouth a favorable habitat for a great variety of bacteria. Oral bacteria include streptococci, lactobacilli, streptococci, and corynebacteria, with a greater number of anaerobes, especially bacteriocytes. So these all the organisms that will include. The mouth presents a succession of different ecological situations with age, and this correspond with changes in the composition of the normal flora. At birth, the oral cavity is composed solely of the soft tissues of the lips, cheeks, tongues, and palate, which are kept moist by the secretions of the salivary gland. At birth, the oral cavity is sterile but rapidly becomes colonized from the environment, particularly from the mother in the first feeding. Streptococcus salivarius is dominant and may make up 98% of the total oral flora until the appearance of the teeth. The eruption of the teeth during the first year lead to colonization by S. mutans and S. sanguis. These bacteria require a non-desquamating surface in order to colonize. So the normal flora occupy available colonization sites, which make it more difficult for other microorganisms to become established. Here, it will changes with age. Birth, it will be sterile, rapidly colonized by environment, and around 6 to 9 months, toddlers, teeth eruption, S mutants, S sanguis, colonized teeth, and puberty, bacteriocytes and spirochetes will increase. So functions of oral flora. Here, it will compete with pathogens for colonization and contribute to vitamin synthesis, induce low level of antibody responses, and produce inhibitory compound, which is fatty acid and bacteriocins. Potential pathogen is oral bacteria may cause abscesses, endocarditis, dental diseases, plaque. As mutants, it will carries dental plaque, acid production, and anaerobes periodontal disease, which is gingivitis. Next is conjunctiva. A variety of bacteria may be cultivated from the normal conjunctiva, but the number of organisms is usually small. Asorius epidermidis and certain corniforms are dominant. Staphylococcus aureus, some streptococci, Haemophilus species, and Neisseria species are occasionally found. The conjunctiva is kept moist and healthy by the continuous secretion from the lacrimal glands. Blinking wipes the conjunctiva every few seconds, mechanically washing away any foreign objects including bacteria. So lacrimal secretions, that is tear, also contain bactericidal substances including lysosome. There is little or no opportunity for microorganisms to colonize the conjunctiva without special mechanisms to attach to the epithelial surfaces and some ability to withstand attack by lysozyme. So here pathogens which do not infect the conjunctiva are thought to be able to specifically attach to the conjunctival epithelium by means of salicylic acid receptors on epithelial cells, but this is not certain. So next is urogenital tract. Urine is normally sterile, and since the urinary tract is flushed with urine every few hours, microorganisms have problems gaining access and becoming established. The flora of the anterior urethra, as indicated principally by urine cultures, suggests that the area may be inhibited by a relatively consistent normal flora consisting of Staphylococcus epidermidis, Streptococcus faecalis, and some alpha hemolytic streptococci. So their numbers are not plentiful. However, in addition, some enteric bacteria are and corynebacteria, which are probably contaminants from the skin, vulva, or rectum, may occasionally be found at the anterior urethra. So the here vagina becomes colonized soon after birth with corynebacteria, staphylococci, non-pyogenic streptococci, E. coli, and a lactic acid bacterium named Do, which is named Doderlens bacillus. During reproductive life, from puberty to menopause, the vaginal epithelium contains glycogen due to the actions of circulating estrogens. Here, Doderlens bacillus predominates, being able to metabolize the glycogen to lactic acid. The lactic acid and other products of metabolism inhibit colonization by all except this bacillus and a select number of bacteria. So, the resulting low pH of the vaginal epithelium prevents establishment of most bacteria and the potentially pathogenic yeast, which is Candida albicans. Next is gastrointestinal tract. The bacterial flora of the GI tract of animals has been studied more extensively than any other sites, and this does not consider all the studies of on the colon bacterium E. coli, which is favorite experimental organisms for microbiologists, geneticists, and molecular biologists. Here, the composition of GI flora differs between various animal species and within the individual's animal species. In humans, the composition of the flora is also influenced by age, diet, and cultural conditions. So, the following table here, the distribution of some common intestinal bacteria in various animal species, including human. In upper GI tract of adult human, the esophagus contains only bacteria swallowed with saliva and food. Because of the high acidity of the gastric juice, only a few bacteria can be cultured from the normal stomach. So, in stomach, few bacteria, which is acid barrier, mainly lactobacilli, proximal small intestine, which is sparse gram-positive flora, and distal ileum, which is dense flora, including coliforms and bactericide. In large intestine, colon, it is densely populated into 10 to the power of bacteria per ml feces. Anaerobes dominate bactericides, bifidobacteria, and E. coli present but outnumbered by anaerobes, which is in 1000:1 ratio. Infant versus adult gut flora. During birth, it is sterile, colonized after first feed. Breastfeed infants, bifidobacteria, which is dominant, less than 90%, and bottle feed infant, which is mixed flora, anthrax, bactericide, and clostridia may be included. Role of normal gut flora, it will synthesize vitamin, example vitamin K and B complex. It prevent colonization by pathogen. It produce antimicrobial substances and stimulate tissue and immune development. Clinical relevance is a disruption. It will lead to infection. Example is C. difficile, colitis. Gut flora linked to nutrition, immunity, and even mental health. So summary for biochemical test. Biochemical tests used to identify bacteria based on enzyme content and metabolic pathway. Catalyst test, it will differentiate Staphylococcus positive and Streptococcus negative. Coagulase test, it will detect pathogenic Staphylococcus aureus, plasma clotting. IMVIC test, which is indole methyl red Vox-Prosker citrate, it is useful for anterior bacteriae. Urease test, it detects urea hydrolysis. Example is protease. Oxidase test, it detects cytochrome oxidase, which is positive in Pseudomonas and Neisseria species. Gelatin liquefaction test, it detects gelatinous enzyme. Nitrate reduction test, it detects reduction of nitrate into nitrite, ammonia, or nitrogen. Sugar fermentation test, it detects acid and gas production. Motility test, it helps in hanging drop or semi-solid media. Oxidase fermentation test differentiate oxidative versus fermentative metabolism. Application include for all this clinical diagnosis, species differentiation, food and environmental microbiology. For AST, which is antimicrobial susceptibility testing, it determines if bacteria are susceptible, intermediate, or resistant to antimicrobial, that is drugs. Drug classification by origin, that is natural, semi-synthetic, or synthetic. Spectrum, broad spectrum or narrow spectrum. And mode of action to cell wall inhibitor, protein, nucleic acid, which is either DNA or RNA, and metabolism inhibitor. Resistant mechanism is mutation, horizontal gene transfer, human misuse, accelerate resistance. Definitions of MIC, MBC, susceptible, intermediate, and resistant categories. Methods include agar dilution, broad dilution, micro dilution, diffusion by Kirby-Beer method, gradient diffusion, which is nothing but e-test. Automated system include YTAC, Phoenix, and Microscan. Superbugs, all this include and control lab technician, infection, lab detection, infection control, hand hygiene, rational antibiotic use. For normal flora of human body, mixture of bacteria, fungi, protozoa, archaea regularly present at anatomical sites. Skin, these are the sites for normal flora: skin, respiratory tract, oral cavity, conjunctiva, urogenital tract, gastrointestinal tract. For skin, all this bacteria will include Staphylococcus epidermis, Micrococcus, Corynebacteria, Staphylococcus aureus in nasal caries. For respiratory tract, upper respiratory tract include Staphylococcus epidermidis, Corynebacterium S. aureus. For lower tract, it will be sterile. Oral cavity, rich flora, Staphylococcus mutans, it carries dental carriers, anaerobes, periodontal disease. Conjunctiva, only few organism. It is protected by tears and blinking. Urogenital tract, urine will be sterile. Vagina, it will be dominated by lactobacillus acidophilus in reproductive years during pregnancy or after pregnancy. GI tract, largest microbial population. Stomach sparse, colon dense with anaerobes. Functions, nutritional support, immune stimulation, colonization resistance, and risk factors include opportunistic infection, dental diseases, and biofilm association problem. For question part, 3 marks. First, define catalase test and state its principle. Second, differentiate Staphylococcus and Streptococcus using catalase test. Third, what is the principle of coagulase test? Fourth, expand indole, methyl red, Vox-Prosker, citrate and give one application of each. Fifth, define MIC, minimum inhibitory concentration, and MBC, bactericidal. Six, give two differences between broad spectrum and narrow spectrum antibiotics. Seven, name two automated AST system, antibiotic susceptibility testing system. Nine, list two functions of normal oral flora. Ninth, why is the lower respiratory tract normally sterile? 10th, name two examples of superbugs. Questions for 5 marks. First, describe the principle, procedure, and result interpretation of the MI-VIC test. Second, explain the Kirby-Bear test diffusion method of AST. Third, discuss drug-resistant mechanism and their clinical importance. Fourth, write short notes on MRSA and VRE. Fifth, explain urease and oxidase test with example. Sixth, discuss the normal flora of the gastrointestinal tract. Seventh, explain the clinical importance of vaginal flora. 8. Describe the role of skin flora in health and diseases. 9th, explain nitrate reduction test and its interpretation. And last 10, discuss the benefits and risk of normal human flora. So with this, we'll cover first topic of systemic bacteriology. In next class, we'll see gram-positive and negative bacteria.
Fantastic, madam. I think today you took more stress, I think. The
No
class, I
issues,
think.
sir.
Yeah.
For second year, no, in detail, I'm trying to
Yeah.
explain everything.
Detailed class we need.
Yes,
And
sir.
mainly, madam, here one topic is there, no, regarding MIC and MBC.
Yes, sir.
So
Minimum.
even doctors get confused about that MIC
Yes, sir.
and
Yes,
MBC.
sir.
Yeah.
But it will be very
Okay.
useful for practical purpose
Yeah.
students. Yes, sir.
I request you to have separate class for that, I think. So I think that
MIC
it
and
will be.
MBC.
Yeah,
All
madam.
right,
You
sir.
can have it, so that no students also get understand
Yes,
about
sir. Yes, sir.
what is the
Okay,
best
sir.
MIC.
This.
Yeah.
Okay, sir.
Thank you,
This
madam.
this
Thank you
part,
so
no,
much.
MIC and MBC, sir.
Yeah.
All right, sir.
Well, if they
Okay.
understand about MIC and MBC, so I
Yes, sir.
think they can easily monitor the sensitivity, intermediate, and what is resistant,
Resistance. Yes, sir.
and
Yes.
they can easily give
Mm-hmm.
the results of this
Mm-hmm.
one antibiotic sensitivity test.
Antibiotic. Yes, sir.
Yeah,
Yes,
yeah.
sir. They
It
do
is very
the
nice.
essays, but they are not able to differentiate between this susceptible, which
Yeah.
is intermediate and
Correct.
resistance.
And which
Yes.
drug exactly useful for a particular bug, yeah,
Bug,
bacteria.
bacteria. Yes, sir.
Right.
All
So
right,
very
sir.
nice, madam. Thank you.
Thank
Thank you
you,
so
sir.
much.
Thank you, sir.
Shall we wind up, madam?
Yes, sir. Yes, sir.
Yeah, thank you. Thank you
Yes.
so much.
Thank